Okay guys, so today we'll be going over cellular energetics, which is unit 3 of AP Bio, and today we'll be only able to cover enzymes. So before learning about enzymes, we need to know the first and the second law of thermodynamics. So the first law states that energy can neither be created nor nor destroyed. So this is my attempt of making an explosion, but I'll just give you an example. So when we're talking about the ecosystem in our daily life, so there's a tree, there's apples there, it has a lot of organisms, it has one sun which is shining light, and then there's humans, and then there's animals. I'll just make a rough diagram of an animal, and then there are so many plants. So what we notice here is that the sun creates light and heat energy through nuclear energy. So it converts from it is so the sun gives us light and heat energy. So when this comes to us, the plants convert light energy into chemical energy, and this chemical energy is stored until one of these apples is eaten by a human. And what happens here is this this chemical energy that was stored in the plant, and for this example in apples, this chemical energy is turned into mechanical energy, and this mechanical energy conversion is not 100%. So it means like about 10% or maybe 20% is used to make up mechanical energy, and the rest is lost as heat energy. And for animals, it's the same process, and for plants, it is the same process. So what we're noticing here, it's that energy is being converted into one form into other. So it's like a cycle. So and we imagine the sun as an unlimited source of energy, and we do that because the sun will eventually run out of energy, but it'll take a lot of time. So in our current lifespan and in our current generations to come, we see it as infinite. So what we're noticing here is that energy is only converting its form. So we have the first law which states energy can neither be created or destroyed, meaning it cannot be created out of nowhere and destroyed from in some form. It can only be converted into one form or another. The second law states that entropy increases whenever some reaction takes place. So entropy you can imagine as the disorder in a system, and by disorder we mean the randomness. So for example, we have these balls in a box, and then we have some red color balls, and this is actually pretty low in entropy, because we see these balls are like organized in like 4 by 2 and color wise. But if we want to look at a box with balls with high entropy, we look at balls like this, which are like all over the place, and then some red balls. And now in this system, the entropy is actually very high, because the balls are not organized in a 4 by 2 way, and they're not arranged in color according to that color. So we see the entropy in this system is actually very high. So we see that any system is like a collection of smaller systems. So any system will always increase in entropy. It will tend to go towards higher entropy. So now that we've got our thermodynamic laws out of the way, next we need to know an important concept which is known as activation energy. So let's take a normal combustion reaction for example. Let's take the combustion of ethanol. So that gives us CO2 plus H2O. So we know any combustion releases energy. So energy is lost. Energy is made it here, and by made we mean converted from chemical to heat energy. So in order to achieve the combustion, in order to achieve combustion, we need to supply some form of energy by heating ethanol, and this energy is first transferred to the bonds in the ethanol molecule, and the energy is used up to break the bonds, and which breaks all the bond in the ethanol molecule. So we see this blue line as the activation energy, which we denote by E with a subscript A, and this is the activation energy. And this whole energy wait this whole energy is the change in the energy between reactants and products, which is called enthalpy, but we don't need to go into this. However, we need to go into exothermic and endothermic reactions. So exothermic basically means you're releasing energy. Exo means energy is exiting, whereas endo means energy is entering. So let's take an example of a combustion reaction, which is an example of an exothermic reaction. So we see, and what this graph denotes is that the reactants have a higher potential energy than the products, and what that means is the reactants store a greater amount of energy than the products, which makes sense if you think about it. The difference in energy which they have is released into the atmosphere in some sort of energy, mostly heat in combustion reactions. So this means that ethanol has a higher potential energy as compared to CO2 and H2O. H2O. So you can think of it this way if you don't understand it yet. So there's a bridge here through which water is falling down. So water is going like very fast in this direction, but when it like collects here, is more or less remains stagnant and stable if it's like going into a lake. And but if it's going into a river, that means it's still not stable yet and it needs to go to the sea. So in this case, we see that the lake down the waterfall is much more stable than the higher energy river, which is dropping the water, making it a waterfall. So now let's talk about endothermic reactions. If we're talking about an endothermic reaction, we can talk about the reaction of iron and sulfur, which gives us ferrous sulfide. And if you've ever seen that, you basically just heat the mixture of iron and sulfur and it gives you ferrous sulfur. So you add heat to make this. So that means that this is at a higher potential energy than this. And you can basically think of the reverse. It's going to a higher energy form, meaning the water in the lake is going to the waterfall I mean, I know that doesn't sound plausible because the water needs to overcome gravity, but it's just an analogy to help you understand it better. So now that we've gotten the concept of activation energy out of the way, we're going through enzymes. And enzymes are basically chemical, are basically biological molecules that lower the activation energy. So let me just show you the graph of that. So this is a graph. This is energy on this axis, and then we have time in this. So imagine if this is the without without catalyst, without enzyme, this would be the reaction with enzyme, and you can see a difference in the energy required to initiate the reaction. And what this basically does is it it breaks the bond in some way so that it's easier for the products to be broken down or to be made up. And some important things to note about enzymes is that one, they don't change the change the reaction they don't they aren't changed they don't change into another molecule during that, which means that they can be reused many times. Broken isn't appropriate, so changed. And they decrease EA, meaning activation energy. They they make it easier for the reaction to occur. Easier for reaction to occur. So when a substrate, the molecule that is to be changed, we call it a substrate, when it interacts with the enzyme, it bonds into the shape of the enzyme, as you can see, like the hexagonal like type of area here. So it basically like fits like a puzzle here, and then it breaks the enzymes into two molecules, or in some cases molds it into two. So this is that. And one more important fact to know about enzymes is that they are very specific. So I know many of you guys must have played some sort of puzzle at some point, and if you've played any sort of puzzle, you would know that they are very specific in what area they will be bound to make the puzzle make sense. Meaning, if you have some, I'm gonna select this. Yeah, if you have, sorry, if you have some other type of puzzle here, if you have this puzzle, it will not fit into this for obvious reasons. So this means that enzymes work on a very process, specific process or specific substrate. And this helps so that an enzyme, maybe lactase, doesn't break down glucose. So lactase can't break down glucose. And that is important because if lactase were to break down glucose, our body would not function properly. One other important thing to know about enzymes are sometimes they may change their shape to allow some other substrate to bind with them. So here we know that this doesn't fit with this because this doesn't represent the shape. If the shape would represent maybe like this, then it would fit. But here we see that the substrate changes its shape to make the substrate bind to it, and this is called an induced fit. The second last thing we have is inhibition. So we have three types of inhibition to control the reactivity, not reactivity, but to control to regulate the activity of enzymes. There are three types. One is called the competitive inhibition, non-competitive inhibition, and third we have feedback inhibition. And what all three does is inhibits the enzyme to form any type of bond with the substrate in order to slow down the reaction or stop the reaction if it's already occurring too much or too many times. So here this is a form of competitive inhibition. So we see an inhibitor which binds to the active site, and I forgot to mention this, but the site where the enzyme bonds is called the active site, as is mentioned here. So here we see that both the substrate and the inhibitor are fighting for the active site. However, in the non-competitive inhibition, we see that an inhibitor may not need the active site to inhibit the process. It needs another site which is oftentimes called the allosteric site. This helps in changing the shape of the enzyme so that this no longer fits in the enzyme. So basically both of these inhibition block the binding of the substrate into the enzyme. And lastly, we have a feedback inhibition, and what that basically does is, so for example, I'll take 3 types of enzymes and 5 types of products. So A goes through enzyme 1 to give B, which goes through enzyme 2 to give a product C, and I'll not make it so much, I'll just make it so that it comes back. So what happens here is, I'll just make the enzymes in another color. So 1, 2, and I'll make the molecule C in another color as well, because it's important to note that. C. So what we see here is that this C molecule is actually a competitive or non-competitive inhibitor of enzyme 1. So if C is produced in excess, it basically stops the enzyme 1 from forming B. So that means B will not form 2, and enzyme 2 will not form C. So this basically regulates the amount of C that is produced in the whole reaction, and B as well. So this is important if our body is producing an excessive amount of a molecule. I'll just give an example, 1 second. So in some cases, if C is produced excessively, it may cause harm to the body. But since C inhibits A or inhibits 2, this prevents the molecule C from being formed, since A will not form B and B will not form C. So this is important to know. And lastly, we have reaction coupling. Reaction coupling. Since we know that reactions are of two types, exothermic and endothermic, exothermics release energy and endothermic need energy. So sometimes what happens is exothermic is coupled with the endothermic reaction to use the energy released by the exothermic reaction. So I'll just give you an example. So for example, A to B is an exothermic reaction which produces ATP, which is basically the energy currency of the cell. And here we have, let's say it produces 10 ATP, and B and C produces D, but it needs 5 ATP. So what happens in this is A produces B since it is energetically favorable, and it produces 5 ATP. And what this, I'll just make it 5 so it's illegible. 5 ATP. And where these 5 ATP goes is it goes to supply the C to D reaction. That means 10 by 5 is equal to 2. So that means this whole reaction can occur 2 times and it can produce 2 Ds, and in the presence of 10 ATP. So these ATP are basically supplying this reaction, and ATP is basically the energy powerhouse of a cell because it has 3 phosphate groups binded together, and the full form of ATP is adenosine triphosphate. So tri meaning 3 and phosphate meaning 3 phosphates. But the problem is these 3 phosphates don't want to be together. So when one of the phosphate is ejected, it becomes ADP, and meaning an adenosine diphosphate. So these diphosphates are basically relatively stable together, and the breaking of the third phosphate releases energy. So that is how adenosine triphosphate is the energy currency of the cell. So now that we've gone over the enzymes and the basics of energetics in this new video, we'll go over the process of photosynthesis and respiration in the next videos. So stay tuned. Thank you.